Okay, Anthony, we're waiting on you. Okay, I'd like to comment on the attendance. So all the students who came willingly, hooray. And I'd also like to comment on the students who came out of some sort of fear factor. I've heard that a fearful email went around. The flogging shall continue. So it's a great pleasure to introduce Ben Hayden, whom my life has sort of intersected accidentally and unbeknownst to me. I've been a fan of his work for a long time. He's now at Baylor College of Medicine, where a whole crew of the what we call the seeding pioneer axons that came from in the neurosurgery department. He did his undergraduate degree at Rice University in chemistry and linguistics, the obvious starting point for neuroscience. Went to Berkeley and worked with the famous Jack Gallant and did some interesting work there, I think in V4. And then moved to Duke University and did a postdoc with Michael Platt, whom we all know, who might well have put a title up like this on his own talks. Michael has always been interested in naturalistic neuroeconomics. And then from there to the University of Rochester for, I think, about 6 years, and then on to University of Minnesota, and from Minnesota to Houston, Texas. His work is actually extremely broad. There's a theme of the cingulate cortex, cognitive control, and things like that. But I think he's going to talk to us today about the impoverishment of our experiments and how to do more naturalistic settings. So I've been trying to get him to come for about 4 years. And so I'm happy the pandemic hit, I think. So it's happy to have you here. Welcome.
Yeah, thank you very much. All right, is this microphone working? I'll tell a story about our lives intersecting. I told you this last night, but I'll tell everybody else. I'm a sophomore in college, and I have these two really close friends. Run into them in the hallway. What are you doing this summer? Oh my God, we found this really amazing guy across the street at Baylor who's doing this just amazing stuff. They described Reed's research program. And I was like, this is the most amazing thing. I was looking for jobs in like chemistry, biochemistry at the time. And I was just like really jealous. I was just like, why do they get all the good ideas? And I just have this boring, biochem stuff that I was thinking of. Oh well. And then like an hour later, I had this epiphany. I was like, wait, I could also start looking for interesting stuff too. And that's when I really started doing research on, let's say, cognitive neuroscience, high level, flaky neuroscience that I've been doing for the last 25 years. And Aaron and Scott were the kind of the guys who really pushed me in that. And they wound up, I think they were in your lab for the summer. I don't know if whatever happened to them, but yeah. So I was at Minnesota until about a year and a half ago. Now I'm at Baylor College of Medicine. This is the photo from the website. And I moved there really with a big shift in my research focus. So since the day I started graduate school in the year 2000, I was doing monkey physiology. I was doing cognition. So I was doing attention, working memory, decision-making, and then really with a big focus on economic decision-making has been my big focus for the last 20 years. Still doing a lot of the same stuff at Baylor, but with a much bigger focus on humans. So I'm actually in the neurosurgery department at Baylor, and we're actually doing single unit, single neuron recordings, but with different species, but getting really excited about all the different stuff we can do. So I'm going to talk about some of the stuff that we've done in monkeys over the last 5 years, but I'm starting to weave into my talk some of the human stuff. So I'll make that clear to you when I switch species and hopefully try to explain some of the benefits of going to humans. So basic stuff that we've been doing for 20 years, and also several people in this room have been doing neuroeconomics. So basically the broad category of my research, the core focus of my research is how do we make a decision? If I ask you, do you want to buy this cupcake for $5? You have to assign a value in your brain to both of those things and decide which one is higher. And the question that my lab is interested in fundamentally is how and where in the brain do you assign that value? How is that value represented? And how does the comparison take place? And the relevance of that is that sometimes you would assign a higher value to a cupcake, but if you're trying to eat healthier, you might nonetheless choose the broccoli instead because it's a healthier option. So there's, so kind of the core focuses of my lab are choice and self-control. And I'm going to talk to you a little bit about that. Kind of tell you a couple brief stories in that broader framework today. Within that world, there's a lot of people nowadays who do decision making, executive control, self-control. Our focus in the lab has really been on trying to make things more naturalistic. Basic gist of the argument on naturalism is it's a real pain in the ass. The analysis is a lot harder. The experiments are a lot harder to design, but it's worth it because it gives you some extra stuff. Fundamentally, there's a validity benefit. This is our lab's official quote, is a quote from Tinbergen, who says, ethology, which is studying behavior, is interviewing animals in their own language. So you're not forcing them to come over to you and do your experiment and hope they understand it the way you want them to. You're meeting them on their territory, and you're trying to understand how they think about the experiment, and there's going to be benefits to that. Here's some of the benefits. I'm not going to read through this whole list, but it's basically a whole bunch of different things. And I'm going to actually, rather than read through the list, I'm going to illustrate this with a couple stories today. So really, I guess intellectually over the last 5 years, we've gotten really interested in kind of throwing away, maybe not throwing away, adding to the classic paradigm of the binary decision. There's this and there's that. There's the cupcake and the $5 that you're choosing between, and moving to what happens when you think about decisions in a continuous sense. What happens when you're playing Pac-Man and you have a decision every moment of Pac-Man, which way you want to turn, do you want to turn around, do you want to go backwards, and how do we integrate that type of thinking into the neuroeconomic framework? So I'm going to tell you about that today with the task that my lab has been playing with for about a decade that we love, and that's kind of one of these sort of never-endingly fascinating tasks. We call this the Pac-Man task. One thing I love about this task, compared to like, I don't know, a Wisconsin card sorting task or something, I'm just going to put this on the background. I'm not even going to tell you what the task is, and you guys probably can figure out what the monkey's job is just by watching this. All right, here's our monkey, blurry. This is from in the dry side of the lab. The monkey's using a joystick here. In case you haven't figured it out, he's controlling this yellow circle. He's trying to capture these squares. The different squares have different juice amounts attached to them. And he's trying to avoid the triangle. If he gets hit by the triangle, he plays a punishment, gives a punishment. So it's a decision-making task only in the absolute, I would call it a decision-making task. I don't think anybody in the Society for Neuroeconomics would consider this a decision-making task, but I consider it one. At every moment of the task, here, I'll play this video one more time. The monkey has to decide which of any infinite number of directions he can head. But then there's also a meta decision. When there's multiple preys on the screen, he has to decide which one he's going to pursue. And if there's a prey and a predator he's trying to avoid, he also has to decide, how do I want to pick a strategy that's going to allow me to integrate pursuit and evasion at the same time? So, this is kind of outside the classical realm of what neuroeconomics is, but I consider it kind of much more valid, much more generalizable to the kind of things that we do in the world. In fact, this is sort of motivated by the fact that monkeys, as primates, we kind of evolved these arms primarily, according to some theories, to catch bugs and eat them right in this space, right? That's kind of the most delicious thing we could do with our hands. That, according to some theories, drove the evolution of our hands and our forward-facing eyes. All right, here's the task. 5 different prey types, 5 different predator types. Some trials have 1 prey, some have 2 preys, some have 2 preys and a predator. Here's just some data. You don't really have to pay too much attention to this. The whole point of this, does this work? Yeah, the whole point of this is that the monkey sampled the space pretty evenly. It's not perfect, right? And so we have to deal with that in our analyses. And I'm not going to spend too much time talking about that, but it's not that the monkeys just go into one corner and always capture the prey. They sample it pretty evenly. And so that's going to give us pretty good purchase on the analysis. And this is just like a typical neuron. This is where the spikes occurred for that particular neuron. We're going to start by looking at a part of the brain called the dorsal anterior cingulate cortex in monkeys. It's the dorsal segment of this region that generally in humans we refer to as the anterior cingulate cortex. We could basically say they're the same thing. I'm going to be really sloppy today and call them both. It's basically sort of here on the middle part of the monkey brain. This is the monkey brain, if you're not familiar with it. Rostral to the central sulcus on the medial wall. And it's generally, in the literature, the theories about its function involve things like conflict detection, like when you're doing a conflict task, it's going to have big activity, value representation, tracking the outcomes of things, so monitoring, helping you choose, helping you monitor the outcomes. If I'm going to say one thing, if I'm going to convince you of one thing, this is the most important slide. If I'm going to convince you of one thing today, is this big idea. In naturalistic neuroscience, as you move from, at least from, let's say, decision making, as you move from simple decisions to more and more complex decisions, as you have in this Pac-Man task, I'm going to show you data very soon, I promise. The right foundation stops being microeconomic theory, and the right intellectual foundation starts to be engineering control theory. So hopefully here at Virginia Tech, that won't be like a very shocking or offensive idea, but you go to other places and people are like, oh my God, this is a whole bunch of new math I have to learn. The bad news is, if you don't know it, you got to start learning it, because as we make the tasks more complicated, this is really going to be the foundation of the stuff we do in the future. So, all right, here, let's just go straight to the data. So this is a really complicated slide, and I apologize for that, but it's cool. It's really cool. So I'm going to walk you through it. So each of these little rectangles is the screen. So remember, the monkey's controlling a joystick, and the monkey's looking at the screen, and it's the computer monitor. All right, and so these are just a bunch of different copies of it. And the z-axis on these plots is the firing rate of a particular neuron. So this is just the first session, second cell that we had. And when the monkey's avatar, when the little yellow circle, when the Pac-Man is over here, this neuron fires a lot. And also when it's over here, this neuron fires a lot. And when the monkey is over here, this neuron basically doesn't fire at all. Okay, so what this is sort of like, think about this like a place cell. Think about this as an analysis that's looking for place cells here in the anterior cingulate cortex. So if you guys probably know, the Nobel Prize, like 20 years ago now, went for discovery of place cells. These are associated with the hippocampus. We have grid cells in the medial entorhinal cortex. There's a graduate student, Kia Hardcastle, who is in Lisa Giacomo's lab at Stanford, and with Surya Ganguly, they did this really amazing study in 2017 where they said, okay, but what if there's spatial information in these neurons that's not clean, like a place cell is? What if it's just sort of in some really weird, random cloud-like shape? How would we know that for sure and not just confuse statistical noise with that? And so they developed this method, and what they found is something I think really interesting that the field hasn't really digested yet, which is that these medial-entorhinal grid cells are really just like one particular degenerate case of a much larger category of neurons in the medial-entorhinal in the rat, that most of which are not clean and elegant like the grid cells, but still convey a huge amount of information. Really cool result. We took the same method and we just said, hey, people have been kind of looking for place cells in anterior cingulate cortex for a long time. Maybe if you really squint at the data, you could find something that sort of looks like it, but they're not really finding it. Well, what if we took this more general, more liberal method that can still detect things that don't look clean and elegant and can find stuff that looks like this and count it as real by comparing it against some cross-validated sample, but it's real here? Will we find neural correlates of place here in the anterior cingulate? And the answer is yes. We find that neurons robustly represent where the monkey's avatar is. Remember, this is a virtual task. This isn't a real task. This is virtual. The monkey's using a joystick playing a video game. Also where the prey is, also where the predator is. So when I say that moving to naturalistic neuroscience has a lot of value, this is what I mean, is that all these theories about ACC are like conflict and choice and monitoring. None of them were like, oh, it's a map, right? But here's a very rich map. And this isn't like a subtle thing. This explains more of the variance of the firing of these neurons than value, conflict, anything else that we look for. The best case scenario, you take all these cognitive variables and compare them to the map. The sum of these cognitive variables is roughly equal to the percent variance explained. So this is a huge driver of effect that we were never able to see because we never had a naturalistic task before. Yes?
Intensity of the reward or the punishment administered in a dosage type way change what is up?
Yes, except not in a dosage type way. The real answer is this is kind of a complex nonlinear mixed selectivity. So it's going to encode the reward expected, and that's going to interact in a really statistically complicated way. Really what we're doing here is we're looking at this really complicated manifold in the high dimensional space, and we're projecting that manifold here onto the single dimension of place. So, but it's not like, I wish I could say, yeah, reward scales this by some multiplier. It's more complicated than that. But the basic gist, I think, of what you're asking, yes, it's true. Reward is in there. Punishment associated with the predator is also in there. So that's allocentric coding. You can do egocentric coding. Egocentric coding is basically, imagine the monkey's heading, whatever direction the avatar is heading is north on this plot. So z-axis here is firing rate again. So when their prey this is the self-prey interaction plot. So when the prey is over to the monkey's right, so if the monkey's heading north, then the prey is over on the east. If the monkey's heading east, then the prey is on the south. If the monkey's prey is over on the monkey's right, and they should probably turn to go in that direction, then this neuron has a high firing rate. And but only if it's far away, not if it's close by. So there's this very rich repertoire of information here in these anterior cingulate cortex neurons. Among other things, I think this helps us answer the question, why are there 300 million neurons in the anterior cingulate cortex? If you think of this neuron, this part of the brain as somebody whose job is to represent values, that takes, what does that take? 1,000 neurons maybe? Represent conflict, maybe that's another 1,000 neurons because there's some noise. There's still like 300 million minus 2,000 neurons left. There's got to be a much richer repertoire in these neurons than just what's going on with these low-level cognitive variables that have taken up 98% of the literature on this part of the brain in the past. I'm not going to walk you through this. All right, I'll just say one more thing about this result, which is if you're thinking about decision making in terms of control theory, one of the things you're going to naturally ask, and I'm sure all of you would if I just gave you another 5 minutes to think about it, is can the monkey predict the future? Is the ACC going to help the monkey predict the future? So in this task, in this prey pursuit task, the maximum speed of the monkey is set to be about like 15% faster than the maximum speed of the prey that he's pursuing. So the easiest thing the monkey could do is just point the joystick wherever the prey is, and he's guaranteed to catch it within the time limit, which is 20 seconds. He'll catch it in about like 9 or 10 seconds. But if the monkey wants to move on to the next trial just a little bit faster, he can do a little bit of mental computation and predict where this thing is going to be in the future and head in that direction. Okay, so if this is the monkey and this is the prey right here, right now, current position, you could just point right at the prey and you will catch it, guaranteed. If you are a little bit smart, you can predict where it's going to be in the future and head there, and you will catch it a little bit faster. If you're just going to do the lazy thing of going where it is, but there's some lag in the system, you're going to head to where it was in the past, and you won't ever catch it. So we can look at the behavior, do some really fancy math. It's all in this paper that my star grad student, Michael Yu, wrote. And basically what we can find is that monkeys, I'm not going to show you too much, is just say monkeys tend to look into the future in this task. They head to where the prey is going to be about 700 to 800 milliseconds in the future. So they're doing something smart. This is actually research that was funded by the Templeton Foundation. The title of the proposal was, "Do Monkeys Think About the Future?" This was the Templeton Foundation is always like, what is the big picture thing? And they were like, what about thinking about the future? So I wrote a grant called "Do Monkeys Think About the Future?" And they gave me some money, and this is what led us to think about this. Yeah, monkeys think about the future. They're going, and there's some interesting stuff here. I don't even have figures to show it. I'll just say anecdotally, when the prey is more wiggly, then the monkeys look into the future less. When the monkeys start this task on day one, their tau is 0. So they're just following the prey. On about 3 months, their future prospection stabilizes at this value of about 700 to 800 milliseconds in the future. And you have about a linear increase across those 3 months. And then it stabilizes. So we ran these monkeys for multiple years on this task, and it stabilized there. We have pretty high confidence in that. But there's this learning. Monkeys can't do this instantly. It takes a few months to do it. I'll just say humans do this within less than 5 trials. Humans can, there's a qualitative difference between how humans handle this task. If you said, and anybody of you, I think, would do this. I say, here's how the task is going to work. You've never seen this task before in your life. You're like, okay, I think I can do this. You can do it. You can do it really well. And within the first 2 or 3 trials, you're already better than the monkey is after like 2 months. The monkey's better than you after 3 months, but if you had 3 months, you would be even better than that. So this is part of what's making us think, hey, maybe this is something different between humans and monkeys. I'm going to skip this. This is basically repeating points that I've already made. All these different variables are here. They're very rich drivers of activity. This is just the percent of neurons that encode all these different variables. Neurons are not specialized for certain things. There are no such thing as self neurons, prey neurons. It's just all giant, as I said, giant, enormous, complex manifold where everything is encoded everywhere. That's interesting and important. It's important for understanding it. It just shows that we shouldn't naively expect there to be self and other neurons. Here's the data showing that there's also a map of future space. So when the monkey is actively predicting the future, there is also an explicit representation. Now, using the same mapping method that Kia Hardcastle developed, we can show that there is a representation of where the prey is going to be in the future. So if the prey is here right now, sort of in the middle of the screen, this neuron fires a lot. If this prey is heading down this way and it's going to be over here in the near future, then this neuron fires a lot. This is the same neuron. So it has different maps for current and future position of the prey. Okay, so this is just mapping stuff, really cool mapping stuff that you could do in ACC.
Have been tested over a fairly long period of time, because at that point they probably have a mnemonic representation of the range of possibilities, the spatial probability of where things are going to be. So does that mean that this is more of a plasticity memory representation, or is it an acute ongoing spatial representation potential?
It sounds to me like you're making a really compelling argument for why we should do this in humans in the EMU who've never done this task before, so that we can answer that question. Yeah. So if I didn't have any human data, I would say that's a really great question. We have no idea what the answer is, but I'm going to show you some human data in about 1 second, where the answer is, yeah, that is a worry, but with these humans who've never done it before, we get almost the same data. But I'm going to say one more thing about the monkeys, because this is the new stuff that we're doing. This is actually my wife, back when we used to go to Monkey Island and hang out with monkeys and take pictures of them, just to kind of like prove that I'm a real primatologist, even though I'm not even doing very much monkey stuff anymore. And I was taking the picture of her. She has a bunch of pictures on her computer of me taking pictures of monkeys. The real goal, the real thing you got to do if you really want to understand choice, I would say, and this again makes me a pariah within the Society for Neuroeconomics, is really our behavior is divided into these discrete states. It didn't necessarily have to be the case. It's been pretty well proven by ethologists, by primatologists who go out into the woods. Yeah, okay, this is how it works. And so there's a big push, starting with people like Bill Bialek and other people, to really take behavior of animals and identify the patterns in that behavior is basically large unsupervised learning problems. We wanted to do this, take this ethological approach, this behavioral classification approach, to understand the behavior of monkeys. And so this is work by Justin Fine, who is a postdoc at my lab. He's now a research assistant professor, also working with me. This was like a huge project that took like 5 years of work to do, but it's really amazing. He basically used a control theory approach to look at the behavior of the monkey in this, and basically asked the seemingly really simple question: which prey? I mentioned there's 2 prey on the screen. Which prey is the monkey choosing? Which prey is the monkey pursuing at any given time? Is he pursuing this green one or the blue one? And it's not just which way he points, unfortunately, because you could imagine that you're heading towards the green one's over there, and you're going here, and the blue one crosses your path. We don't want to think that you're still pursuing the blue one. I'm not going to walk you through this because I want to get to the human stuff. But he did a lot of math. It's all in this bioRxiv paper that you can see. And basically, what Justin found is that not only are the monkeys strategies, they're not just pursuing one or the other. The strategy is actually continuous. They can blend their strategies. So if you use enough sophisticated control theory, you can find out that the monkeys have this meta-control problem that they're solving. Not just a control problem, but a meta-control problem where they're trying to decide which of these two preys to pursue and whether to blend the strategy and sort of simultaneously split the difference between the green one and the blue one and sort of go between them. Or maybe there's a predator chasing them and they're trying to both pursue this and evade this at the same time. And so if you do all the math right, and we can validate this using a bunch of extra data that we collected, you can say at any given time what the weight was. So imagine there's only, let's keep it really simple. There's only two prey, and we're just going to call it by arbitrarily say one is a high value prey and one's a low value prey. You can do this in a bunch of ways. It won't matter. Let's take a bunch of times when the monkey switches strategy from pursuing one to pursuing the other. That winds up being the most interesting thing. So because we're going to ask, why does the monkey switch strategy? Why does he switch from one state, behavioral state, to another ethological state, right? So this is like, in an abstract sense, why do you switch from foraging to like grooming your partner? But in a very narrow sense. And what we find is that we can find these crossover points. Generally, the monkey's behavior when they're pursuing a prey is somewhere around with a W factor of around 0.7 or higher. So usually they're mostly pursuing one prey, although seldom at exactly a fully. They're always putting a little bit of weight on the other one, basically keeping an eye on the other thing. And then when they switch, you see this big swoop over here. We have an equal amount of data when they make the other switch. I show this to you mostly just to show you that we can do this. We can recover this from the data. Now we can start asking really interesting questions. Like, for example, what are the factors that drive a strategy change? They're not going to be that surprising. Basically, when the prey, when you're pursuing one prey and the other prey comes nearby, that's when you're more likely to switch to the other prey. When the time in trial gets late and the monkey gets kind of desperate or bored, he's much more likely to switch over to the easier to catch, smaller value prey, things like that. So we're doing basically ethology in this prey pursuit task. Yes?
There are other factors like the speed of the other prey.
Yes.
And the
right.
direction of the other prey.
Yes, absolutely. Yeah, there's a whole bunch of other factors. I'm just kind of summarizing for you some of the ones that jump out at us. But right, there's this whole surface of factors, and they interact with each other also. So there's another interaction between these different factors.
The reward value is held constant.
So the value of catching the prey is held constant. But if you think about it as from an engineering perspective, when the prey is closer to you, it has a higher subjective value because you can catch it more easily. So that varies continuously over time. We have to deal with that. But I think what you're asking is the amount of reward expected from catching it, that's held constant. Now, Justin, of course, now that we finished this bioarchive paper, he's like, let's do a version where we have the prey fluctuate in value over time. So we're gearing up to do that as a sequel in our humans. I'm going to skip this. I'm just going to say that neurons encode this meta-control variable. We also have some recordings in dorsal premotor cortex. We find encoding there too. They don't look that different yet, but we think there's going to be some differences that we see between these two regions. I want to get to the human stuff. I'll just say one more thing about this. At the moment of switch, the thing that this is Tolman's question. What are the determinants of behavior at the choice point? What happens when the monkey switches from one state to the other? Does this sort of classic idea of a ramp of activity that rises to some threshold, and then when you get to that threshold, then you make the switch? Does that apply in any abstract way to the switch between pursuing the green square and pursuing the blue square? And the answer is it does. So if you take the entire population of neurons, project the activity onto a low-dimensional variable that is associated with the time of the switch, then you see this ramp. This is average over time, but this tends to be about 1 to 200 milliseconds. So there's different speeds, and we deal with that by normalizing the time. But in practice, this is a couple hundred milliseconds. You see a ramp of activity. This is not firing rate on the y-axis. This is the projection. So basically, it comes to resemble this state. This is like dynamical system stuff. There's a state associated with making the switch, and it comes to gradually resemble this over about 200 maybe 300 milliseconds leading up to that switch, and then it dissipates. So we can see some, in a very abstract sense, some integration process or some gradual rotation process in a high dimensional space as you move towards this switch. So this is kind of what this paper is about. This is actually not even in the bioRxiv paper. This is new stuff that Justin just sent me. Okay. I'm talking about humans. How are we on time? Okay, 11:30, great. I'm talking about humans for a little briefly. I have a new graduate student at Baylor College of Medicine, Asia Ciarconi, who is really fantastic. And so her first assignment that I gave her was to replicate this experiment in humans. And we now have data from 10 patients in the EMU, where we were, we have 13 now in the epilepsy monitoring unit. Okay, so you guys probably all know this, but basically there's patients with very severe epilepsy, and they're going to come to St. Luke's Hospital. They're going to get monitored for 1 to 2 weeks with often, dozens of electrodes in different parts of their brain, especially the hippocampus, the mediotemporal lobe, and the anterior cingulate cortex. Those are the big ones, but we also have some data in a few other regions as well. And they're going to be sitting there waiting to have epileptic seizures because the surgeons want to know where exactly the seizure is coming from so they can either resect that or do some kind of pacemaker in the brain, essentially. And so they're often just bored. And so they're going to come in, sit there, be bored, and we're going to come in and very politely say, hey, do you want to do any psychology experiments? We're not going to say these are things that monkeys were doing a few months ago. We just say these are some really interesting things that we want to see. And what I can tell you is, yeah, they're usually pretty happy to do this. So this is a big benefit to us as scientists, and we're very grateful to them. And so we have them do exactly the same task as the monkeys. Now we're doing some variants on the task as well.
They are not taking any particular medication.
No, they're on a lot of very serious medications, actually, and they have seriously diseased brains. So these are all caveats you have to worry about. They're often clinically depressed as secondary to their epilepsy. They're very distracted. They just had surgery a few days ago. These are all very serious concerns, and I don't want to minimize them. On the other hand, getting unit data from humans is a pretty rare thing. Yes. Sorry.
Why don't you just do it with volunteers? They don't have that particular condition.
Surgeons aren't really willing to drill holes in the brains of people who don't have severe epilepsy, put a bunch of electrodes in their brain. It's not really ethical to do that. Yeah. It would be great if we could. That would be, scientifically it would be more meaningful, but ethically we couldn't do it.
Yeah.
Actually, what's stranger, epilepsy or the willingness to have your head drilled?
Depends how much you pay.
Could you speak to the capture points in the monkey that pile up on either side of the vertical lines? Are they learning to trap the target?
Yes, I think that's what's happening.
Okay.
Yeah.
And why don't the humans do that?
So this is data from our first human, and we've seen this very consistent pattern. I have data from Asia. It's very consistent. So the humans seem to employ a different strategy. And right now, one thing that we're doing, and we're looking for ways to kind of formalize this, anecdotally, the humans have never done this before in their life. They're much better, and they've captured the prey much more quickly than the monkeys do, even after 3 months. Humans seem to have an intuitive grasp of the physics of this, and they seem to do these things that feel intelligent. This is just anecdotally, like cornering the prey. The monkeys don't seem to do that, even though the task is exactly the same. The joystick is the same. As far as we can tell, the humans have this kind of insight. And as you can see, by the end of the 6 months or year that the monkeys are doing this, they do somewhat tend to do this. So these points cluster over here on the side. That happens more often near the end of the session, at the end of the the human traps it. And remember, this human has never even heard of this task before 5 minutes ago, and they're sort of already starting to do this. So it seems like there's some, if you might say, insight or something like that the human has that the monkey just didn't get in several months.
Just a question on the patient population. Where are the epileptic foci? Where are you? So they're mostly cingulate epileptic foci, or is that just?
We take everything. Generally, they're going to be a mediotemporal lobe, hippocampus, medial temporal. Anything that's where the epilepsy was determined to be, we exclude from the data set post hoc. So we don't know at the time. It's only afterwards that analyze the data. Any time that there was a seizure going on, we exclude that data. Or if you're within like a couple hours of the seizure on either side, we exclude that data. So we're trying. These are standard methods in the field. We're trying to avoid diseased tissue as much as we can.
Struck me that if it is medial temporal, particularly hippocampus, that they may have a generalized issue with spatial localization.
Absolutely.
And so, it raises a little bit of a question about how to interpret how they do this.
Absolutely. Although I will say we now have a large data set of healthy humans, people without epilepsy, and we have compared the behavior in the humans to the behavior in the monkeys and to the compromised humans, people in the EMU. The behavior of the humans is basically very consistent, regardless of species. But we do see this really intriguing regardless of health status. We see this very intriguing species difference. So we're just starting to try to figure out how that ramifies. Yes.
So if you're going to answer this, going ahead, just tell me to shut up. But in case not, in the humans, do you see any emergent behaviors? I was thinking of a herding behavior where with the two kind of, getting one to go closer to another and grab them both at the same time, for example.
Yeah. There's not a real well. So right now we're sort of take this ethogramming approach, and we're saying yes, anecdotally we see that. Now, how do you quantify that? How do you prove that? That's really, really hard. But we're working on ways to do that. That's kind of the next step of what we're doing right now. Okay, generally speaking, we get anterior cingulate in the human and the hippocampus. We also sometimes get orbitofrontal cortex as well, and that's also of interest to us. Right now, I think we have OFC data in 4 patients, and the rest of these we have 13. All right, so with all the caveats that this is diseased tissue or the diseased brains, although presumably pretty healthy tissue, we find very similar data in the humans. The data is of lower resolution than it is in the monkeys, because in the monkey we typically get a few thousand trials. In the humans we're really limited and practiced about 30 minutes, sometimes 40 minutes. And so it's this very precious resource, but it's enough to see statistically significant results. Another thing that we're doing right now is we're trying to see, is there any difference between hippocampus and ACC? So if you remember in the monkeys, we were only in this part of the brain, the ACC. And in one slide I showed you, we also have some data in dorsal premotor, which is adjacent to ACC and expected to be not that different qualitatively. Hippocampus is several centimeters away. Hippocampus is the part of the brain where you look for place cells, you expect place cells to be. So we thought maybe we've seen some place cells here, but we really don't. It looks a lot like the ACC. And so we're trying to come up with methods to find any difference at all. And I can say qualitatively, there's nothing that jumps out at us as a big difference between hippocampus and ACC. I'm going to show you a little more detail in 2 slides from now. I'll just say we have a very small number of I think we have 50 neurons right now in the orbitofrontal cortex. Those OFC neurons also look the same. Very important controversy in the history of neuroeconomics. Is there any space information in the orbitofrontal cortex? The general consensus in the field is no, but generally the field does not use these kind of complex mapping tasks where you have to keep track of space in order to do the task. And so what we find is that when humans do this task, we find in the 50 neurons that we have, this looks a lot like the spatial representations we have in these other parts of the brain, including the hippocampus. All right, so here's the kind of summary data. So this is the anterior cingulate cortex of the monkey right here. And this is just a bunch of variables, and I apologize because you would have to know this paper really well to know what this is: position, velocity, direction, velocity of the prey, the position of the prey, the velocity of the prey, things like that. These are the egocentric, these green ones are the egocentric variables. So the relationship between the self and the prey, the angle between the self and the prey are both in there. In the monkey, in the ACC, and the hippocampus, things look pretty similar. These are the human data, and this is the monkey data. Things look pretty similar across species. The general way this game is played in physiology papers is you look for any subtle differences between these things, and you write a whole paper, and you say that's the key to understanding the anatomical difference between these regions. I try to avoid playing that game because I feel squiffy doing it. Generally, what I look at this and I see is like, yeah, there's a lot of similarities between these. We're going to do what we can to tease apart the differences, but it's certainly not the case that like hippocampus has place and ACC doesn't have place. Certainly, it's not the case that there's some enormous difference that jumps out at you and says this is the real difference between ACC and hippocampus. I've never been a hippocampus person before. I'm doing it because we have this human data. I moved to Baylor because we were getting so much ACC data, which is my favorite part of the brain. But the hippocampus comes along for free. The electrodes are in there. So now I'm becoming a hippocampus person. But I don't think it's going to make me very popular in the hippocampus world to say that, yeah, to our eyes, ACC and hippocampus look pretty similar.
Visually guided, do you have any sense of if you were to use a different sensory modality, if you get the same representation of place or an auditory cue? I mean, it would have to be done in a different behavioral task.
Yeah.
It's the same issue of individuals.
So Matt McGinley at Baylor College of Medicine is doing a lot of auditorily guided foraging, and I'm spending a lot of time talking with him and hanging out with him, having a lot of fun with his lab. So far, in a very, I can say to a very abstract sense, very preliminary sense, it looks kind of similar, but it's very early days in that type of research. So my speculation, in theory, these are kind of abstract multimodal areas very far from the sensory input, so they should be, as long as you're doing the navigation, it doesn't really matter how you learn that, but that's just a guess though. All right, I'm going to tell you about the project that really ate up the vast majority of my time over the last, since really almost 10 years now, is trying to solve this problem of freely moving monkeys. So really, monkeys don't play video games. Really what they do is they move around the world like this. This is a cage that we had at Minnesota. We have space identified at Baylor to do the same thing. So I'm working with Jeff Yao, who's a monkey physiologist there at Baylor, and we're going to do tracking there, and we're going to build the same thing there. So we're writing grants to do this also. So we want to really push as hard as we can in this direction of naturalism and moving into the natural world. I'm going to tell you about the experiments that we did at Minnesota and where we're going to go with those. We have the monkey sit in this big room. He is surrounded by 62 cameras following him. We're not using deep lab cut, but you can think of it as being basically the same type of thing. It's a bespoke thing that's basically tailored to the physics of monkeys. That was developed by a computer scientist at Minnesota. That guy, by the way, Hyun-Soo Park, really amazing guy, really fun to collaborate with. He now quit academia, has a company in South Korea, which is where he's from, and he's selling it to daycares to track your children over the day. And he's trying to expand into this country. So if anybody runs a daycare and wants to have this technology, it's extremely expensive. I don't know why people are doing this, but people want to know where their children are. 13 key points, 300 hertz resolution. Very popular apparently in that country. So and he's, I think, having a lot of fun going, not in academia, running a small business. But he helped us build the monkey thing. I think he kind of decided there's a lot more money in children than there is in monkeys. So he helped us finish the papers and then he moved. Yeah. I also put this slide up here. This is the Menninger Clinic, which is a psychiatric inpatient and outpatient clinic that's at Baylor College of Medicine. We are also starting work, and we now have funding from the NIH to do this where we're going to have humans move around the campus. We're going to track them with cameras and a bunch of other things like their phone that tell you where you are in time. Or we're going to start doing a lot of the same neuroscience with these patients that have electrodes implanted in their brain. These are going to be depression, obsessive-compulsive bipolar patients who have electrodes. They have to come here every 6 months to get their parameters retuned, and we're going to have them do this test before the retuning, after the retuning. We get LFP data. We don't get unit data, but we get really cool data from them while they're doing these real life navigation tasks. Because that's new, and that's something we're just starting. We just start interviewing postdocs for that right now. I have no data to show you, but I just wanted to stick a little more human stuff into my talk. These are all the people. This is Hyun-Soo here, and this is Jan Zimmerman, who's still at Minnesota. And this is all the people. There's actually a bunch more people that were doing stuff, but these are the core people that really helped us build this system of monkey tracking. Here's our thing. We were collecting 3 terabytes every hour, and we would do 4 hours of data collection a day. And then we would spend 20 hours processing the data just to clear off the server so that by the next day we could run it again. It was this huge operation. We did this for 6 months for the first monkey and then 6 months for the second monkey. And then we said, now we're going to never do another experiment for 5 years. And we're still writing papers on this data set. There's this exoskeleton. The monkeys are really violent, and they'll often push against the edge of the cage. We don't want even a little bit of vibration. So these cameras are on an exoskeleton. These are machine vision cameras. There's another view of it. We painted this green because the same reason they use green screen in Hollywood. It's easy to segment. Jan designed this whole thing himself. Jan Zimmerman did. We record, but this is basically a gray matter drive. Charlie Gray's drive is 128 independently movable electrodes. And then he built this whole housing thing that also records the data, essentially a tiny little plexon on the head of the monkey. I remember when we moved to Plexon in like 2002 and how great it was. And this Plexon is like this big. No, it's easier to just put it on the head of the monkey than it is to try to figure out how to stream it across the room or to put a wire on the monkey's head. We recorded in 6 brain regions. We have this data set now with over 10,000 neurons. And we've done a lot of really cool stuff with it. So we now have 4 papers that we've written. And we're continuing to do more, and we're starting. If you're interested, for some reason you're interested in this data, let me know, because we keep such a huge amount of effort into this. We want to like share this with as many people who have scientific ideas to do with it as we can. Here's just a video of what it looks like. I'm sorry, it's a little bit grainy, but this is actually the first day we ever did it. This is the monkey. And so he's having a bunch of neurons recorded at the same time. Oh, this is really grainy here. I'm sorry about that. But you can see what's happening as he's doing this task. There's this little screen over here with the light and the computer screen, and he can interact with it. There's another one over here. We spent at least a year playing around with peanuts, M&Ms, grapes, blueberries, frozen blueberries, and we eventually decided that water is the best thing to do. Actually water is even better than juice because you don't have to clean it out. It doesn't get sticky in there. And the monkeys will do more trials more voluntarily just for small squirts of water than they will. They'll be like, M&Ms, I love M&Ms. And then after like 15 of them, they're like, that's all I need. Whereas water, they'll do this for the full 4 hours. The first thing we did here is we did these prefrontal regions, and we started doing these spatial things. You guys are not going to be surprised when I tell you that there is clear spatial maps in all 6 of these regions: orbitofrontal cortex, anterior cingulate, and then these other regions that I haven't made a big deal about today, but we had them for free. So rich representation of space, of where you are in space and where you're looking relative to the room. Again, I don't want to emphasize this. I don't want to be like the guy who's like standing on this corner screaming, but saying, everybody who has these theories about these regions that is a very rich and narrow, impoverished version that doesn't involve space is missing a big picture. That's all I'll say about that for the whole rest of the talk. More interestingly, we took the behavior and we did a essentially unsupervised clustering of behavior. If you imagine the position, so we have these 13 points on the monkey's body. We switched at some point in the middle of the study to 15 or 17 and 17 as the technology got better. So I don't remember how many, 1, 2, 3, 4. Well, anyway, this is basically where the monkey is. If you have the monkey always kind of face the same direction and you keep the head the same place, then you can take all those 13 points, essentially treat it as a gigantic matrix and do an unsupervised clustering on it. And what you do with that, when you do UMAP, what you do with that is you can reduce the dimensionality through UMAP or any other method. It doesn't really matter what. And you're going to get some kind of representation of the space. You can look at you can think of this as a mountain range and draw lines on the ridges between the mountains. And what you can do is you can automatically detect different islands, essentially, different mountain peaks. So basically what you're doing here is you're asking the data to tell you, this isn't neural data, this is just behavior, how many clearly classifiable distinct behaviors are there in the monkey's repertoire over the course of, this is actually 3 monkeys, so this is like a 1.5 years of data collection of the monkeys coming into the cage every day, 4 hours, doing whatever they want, foraging, playing with the task. The answer is there's 59 behavior states. And we can start looking for neural correlates of that. But yes, there's a question over here. We definitely have head position. And doing eye is a lot harder.
There is a spatial map of eye movements that is mnemonic and is,
Yes.
also referring to the animal's positions.
Yeah.
You wonder whether or not it's following this or it's leading.
Yeah, I feel guilty saying this because we got a very small grant from the robotics group at Minnesota to solve this eye tracking problem, and then we never did it. Yeah. In theory, it's doable, but we just got so overwhelmed with this project that we never did it. Basically, it's a spatial resolution problem. So you need a camera that has good enough resolution. We also considered putting eye coils in the monkey and using the cage as a giant Faraday cage. And there was some other solution. We were talking about this again. Jan and I were talking about this again recently with our new cage that we're building at Baylor. We want to do the eyes again. The real truth, I think, is that when you're freely moving, your eyes are mostly looking straight ahead. And so that winds up being a pretty good approximation for where you're looking. But of course, they're not always looking straight ahead. So yeah, we do want to solve this problem. There's also, you could also mount a camera on here. Yeah, so that's what I remember now. Yeah, we're writing a grant for a cage here at Baylor. And we were like, do we want to mount the camera here? Yeah, you could do it, but just a headache. But we don't have it in this data set. All right, I'm running out of time, so I'm going to go really fast and just say, ethologists have been talking for like a century or more about what is the hierarchical structure of behavior. There's been the theories about it. It's pretty clear there is, but quantifying that in a way that has no humans that could potentially bias you has been hard. And so we were able to build this hierarchy of behavior for monkeys. And so we could say, yeah, there's these 59 different behavioral states, and this is how they're organized. I don't know that there's any special wisdom that you get from looking at these. We have another paper that's, it's not the Cell Reports paper, it's in Oxford Open Neuroscience, where we show that the hierarchy changes depending on the context. So if the monkey's doing this task, or if he's just bumming around, no task at all, the hierarchy changes. So we can quantify this, we can do this, although I would not say we have any amazing insights from this. And then the last data slide I have is just to show that neurons in all 6 of these regions encode this. And they're not all exactly the same. The more dorsal you go in the brain, the stronger, the more variance explained. And the more caudal you go in the brain, the stronger. So it's like as you move, as the prefrontal cortex kind of wraps around the genu of the corpus callosum, the more you go in this direction, the weaker the encoding is. And the closer you get to motor cortex, the stronger is the representation of action of state. And why that is, I don't know, but what I will say is we're not seeing big qualitative differences that vary with what anatomical brain area we're in. We're just seeing these really big gradients of function. And that's, I'll give you, that to conclude, I'm going to give you a few thoughts that I have after several years of thinking about these experiments. These are just kind of big picture thoughts. 5 thoughts. Number 1, in naturalistic tasks, firing rate is much, much lower. And yet the information content is about the same. This is like stuff that I was doing in grad school with natural images. The firing rate of visual neurons is lower, but the information rate is the same. This is what we find with behavioral correlates. Why neurons fire a lot less actively in these naturalistic tasks, I don't know, but it's an interesting observation. Number 2, a lot more downtime. You train a monkey to do this play pursuit Pac-Man task. They're just going to sit there. They're going to do that for 4 hours. You're going to put them back in their cage and you're done. In this big cage where the monkey's going to sacrifice a lot more juice, they're going to spend a lot more time just kind of sitting around. Maybe it's because physical movement is harder. Maybe the leisure trade-off is, the balance of those things is different. I don't know why that is. There's just spend a lot more time just relaxing. The monkeys seem to be a lot smarter in these tasks. So training on this, so we did like 3 or 4 different tasks in this big cage environment, like a bandit task and some card sort type tasks. The monkeys seem to learn these a lot faster in this big cage than they do in a chair. So they seem a lot smarter. They seem to be much more like teaching themselves. So it's not like they're just being lazy in this cage. They're like in a different mind state that makes them smarter. So this is great because you don't want to have to walk in there and like move the monkey's hand and have him press the button. it would really suck to do that. And so we kind of got lucky on this. The monkeys do these tasks much more intelligently in this freely moving environment. Much bigger individual differences. I could tell you easily the difference between a 3-year-old monkey, a 5-year-old monkey, and a 7-year-old monkey, just looking at their behavior in the big cage. Very different, very age-specific. Gender differences are a lot bigger. Just individual differences across monkeys. Huge difference. In a gambling task or a card sort task or something, sitting in the chair, I could not tell you the difference. I could not tell the difference between a 2-year-old and a 20-year-old monkey. They look exactly, their gambling behavior looks exactly the same. So I think by moving to the naturalistic world, you uncover a lot of behavioral idiosyncrasies that you don't see in shared tasks. Pretty interesting. Last thought before the end of the talk, because we're almost at the end of the time anyway. We just don't see these brain regions that are the kind of the foundation of modern systems neuroscience. You know, basically the Brodmann areas or the Walker areas. We don't see evidence of those, even with this really fancy recording thing where you have 6 different brain regions, a lot of simultaneous data. It just looks like, you know, there's this big gradient of function that somehow the way that these neurons behave, the types of functions that you evoke are not, there's nothing that changes when you move to the anatomical border in these naturalistic tasks in the way that you might expect from a shared task. So I'm going to just show you this video of the imaging center at Baylor, which you guys might memorize. This is not the imaging center. This is a virtual reality game that we made that we have patients play. So we have them walk around the imaging center when they get their initial scan. And then when they're in the EMU 2 weeks later, they play this virtual reality game where they're using a joystick to move around it. And we're going to do some, you know, hippocampus navigation type stuff with it. We have data from 4 patients. I haven't analyzed it yet, so I can't tell you anything about it. But this is what we now call Camry. This is walking towards one of the scanners. There's a bunch of art on the wall. So the cover task is that they're supposed to evaluate the art. And then 2 weeks later, when they're in the EMU, they got to find the paintings again. And I'm going to stop right there and let you guys go because I don't want to take too much time. Yes.
It's about the humans wandering around. So you guys are going to have humans with DBS electrodes in various places with the wireless.
VCVS. So they're going to, yeah, basically in one place.
In what?
In one place, in the ventral capsule, the ventral striatum. So that's a very specific patient.
For OCD or for...
Yeah, OCD, bipolar, and depression, all 3.
And the wires won't be exposed. It'll be talking, there's some recording.
Yeah, completely internalized. Yeah. So this is a very special patient population that we have access to for, and they had their surgery a long time ago. There's only this one place. So yeah, we can come back and collect the data. Yes.
So I was curious about your prey predator test. You mentioned that it takes longer
for the monkeys to start sort of looking into the future than it does for humans. And I was wondering how much of that might come from just unfamiliarity with using a joystick versus most humans have played a video game or two, so they sort of get how a joystick works. How do you deal with that, or is it just not really a big deal?
It is a big deal. So humans have had joystick training. Most humans now have had since they were kids, right? They played Atari. Atari has been around for 40 years or more. And monkeys, no. But on the other hand, I will say the monkeys have had, before we even start them on this prey pursuit task, we train them on the joystick. That's the hardest part, actually. They basically can learn, once they know how to use a joystick, they can learn the prey pursuit task in less than a week. But it takes 3 to 4 months to train a monkey to use a joystick. And that's like 5 days a week for like 2 hours a day. So they have had copious amounts of experience. On the other hand, it's not like a lifetime of experience that I have had playing Atari and Nintendo and stuff like that. So, you know, how much of a difference can you ascribe to years and years of learning? Those are important questions. I don't want to minimize those questions. These are just things that we're starting to notice as we compare the 2 species. And we're thinking about ways to deal with that.
There's an online question. Could you please elaborate on the specifics of reward punishments for monkeys and how these relate to reward punishments for humans, if any?
Yeah, great question. The monkeys have a metal straw in their mouth and they get squirted their favorite drink, which is usually water or diluted cherry juice. Their punishment is timeout, anywhere between 2 and 10-second timeout, which the monkeys don't like, but don't find that aversive. Humans, the only reward and punishment is points. So there's a huge literature. This is a big concern when I started doing neuroecon 20 years ago. You're probably part of these debates. How important is it to make sure the rewards are matched? My conclusion, my read of the literature is humans are pretty good at just doing stuff for points. If you tell them, this is important, do this. Most humans want to do this. So we're not worrying about that. I did an experiment 20 years ago where we had humans doing a gambling task for squirts of juice in their mouth. We had them get really thirsty, not drink any water for 4 hours, and we got some cool things, but it's not really that different. That's what a lot of people were finding. So yeah, this is another difference between the species. I don't think that's driving it's not driving the similarities we find between the two species and how their brains represent this information, but it's definitely worth remembering that it's not exactly the same task.
Right, well, one more and then I'm gonna let everybody go.
I just wanted to ask in relation to Eddie's question about the muscle memory of using a joystick in the humans versus the monkeys. If you inverted the joystick controls, how do you think that would relate to both the data that you're getting and like the time that it takes to learn the task?
You're giving me horrible headaches just thinking about playing GoldenEye with my roommates in grad school. I can't do with the inverted joystick at all myself, and it makes me feel sick. Car sick to think about it. Yeah, we never done that. Yes, there are things that we could do to kind of make things a little more even for the two species. So these are all things to be concerned about. Right now, what we're trying to figure out, big thing we're trying to figure out is why these brain areas look so similar to each other across the two species. And if there are any species differences at all. But I think we're going to start to untangle those, and when we do, we're going to be thinking about these differences in experience and training.
Thanks again. Let's thank Ben here for that.